Hello, thank you for attending my GLBIO presentation entitled Towards Computing Attributions for Dimensionality Reduction Techniques. My name is Matthew Scicluna. I'm in the lab of Julie Husson and Sebastian Lemieux, and these are my affiliations. To begin, I want to say that the paper will soon be available on bioRxiv, a preprint, and you can see the QR code for a link to the GitHub repo that contains all the code here. The motivation is the following. In bioinformatics, we have an abundance of omics data, genomics data, transcriptomics data, proteomics data, and this data is growing at a rate that's exceeding even Moore's law. Furthermore, this omics data is very high dimensional. For example, genotype data can contain hundreds of thousands of dimensions, so it can be very complicated to work with this kind of data. We can work with this kind of data by using the following assumption. Perhaps our data exists in a low-dimensional manifold, and it only appears to be in a high dimension. What you see on the top right here is a very simple data set that appears in a lot of different applications called iris. And we have three measurements for a bunch of plants, and we plot the three measurements in this 3D space, and then we color it based on the species of plant. So you can see Iris setosa, Iris versicolor, and Iris virginicea. We can also see that these points are clustering based on the plant species. Indeed, we can fit a plane on these points, and we can see that when we project the points onto this plane, they actually look very similar as they did in the 3 dimensions. And this tells us that maybe there's some redundancy in this representation. When we look at this plane in the right angle, we can see that indeed the cluster structure is preserved in the 2D space. It looks very similar to the 3D space. We call this machinery that takes us from 3-dimensional space to 2-dimensional space as a dimensionality reduction algorithm, and we illustrate it as a black box because we're going to keep the contents hidden for now. We call the representations on the right-hand side, the 2-dimensional representations, we call that an embedding, and we can visualize the 2D embeddings using a scatter plot. We can search for patterns in this scatter plot. Unfortunately, it can be hard to know why the embeddings appear as they do. Usually people would use things like enrichment analysis to understand the t-SNE embeddings, but these are post hoc. It's not clear what the justifications are for this. And this motivates this presentation. We would like to develop methods to help us understand why dimensionality reduction algorithms, why the embeddings appear the way that they do. Now, let's get to the background. Different dimensionality reduction algorithms have different strengths. t-SNE is the algorithm we're going to be talking about the most in this presentation. In order to do t-SNE, we first compute the affinities, which is inversely proportional to the distances between all pairs of points. We represent this as a matrix P. And you can see the affinity matrix on the right. We've color-coded it so that points that are close by appear dark brown and points that are far away appear light beige. Now, we're going to initiate the t-SNE embeddings randomly, and then we're going to compute affinities between all pairs of embeddings, and we're going to call that Q. Because the points are initialized randomly, the affinity matrix Q is going to appear like white noise. And we also want to point out here that the t-SNE embeddings are not computed, like the affinity matrix Q is not computed using the same similarity function as P is. Now, during the evolutionary of t-SNE, the embeddings are nudged in a direction such that the embeddings of nearby data points will be close by. And you will see here exactly that happening. And finally, the matrix Q ends up looking a lot like the matrix P, and the Y's are being adjusted such that that happens. To be specific, the update formula you will find below. yi of t is the t-SNE embedding of data point I at time t. And we compute it by taking the previous iteration and then adding this dyi t minus 1 term. And that dyi t minus 1 term, you will see, is basically just a sum between all pairs of points, the difference between p and q, and then weighed by the embeddings, the distance between the embeddings. And we've rewritten that as this phi ij t minus 1. And you will see that reoccurring throughout this presentation. Now let's talk about interpretability. And specifically, we're going to talk about the feature attributions framework. So we have our data matrix. Each row is going to be a sample. Each column is going to be a feature. And we're going to pass this thing into a dimensionality reduction algorithm, black box. We're going to end up with two-dimensional embeddings that we can display as a scatter plot. Then we're going to take some aspects of the dimensionality reduction algorithm as well as some aspects of the overall final embeddings, and we can compute scores, one score per feature. And each of these scores will be different for each individual. So we've only displayed one for this in this case. And a bigger attribution score can be interpreted as that the feature contributes more to the overall placement of t-SNE. Okay, and the particular kind of feature attribution we're going to be using are called gradient-based. There are many examples of gradient-based feature attribution methods. They're usually used to understand the output of a supervised classifier trained to perform some computer vision task, but they can be used in a lot of other contexts as well. And the rough idea here is that if the output changes a lot, then the feature should be important, and the gradient is a measure of that. To illustrate this, I have two simple functions we can look at the gradient of. On the left, we have a constant function. Clearly, f of x is not affected by values of x, so the gradient will be 0, and indeed, a gradient-based feature attribution score would also be 0. And this is what we expect. On the right-hand side, we have a parabola, and for a parabola, the value of the function is clearly dependent on the value of the feature. So we would expect to have a positive or negative gradient. And indeed, that's exactly what we have. Note, though, that depending on where you take the gradient, you could also have a gradient of 0. This is an important limitation of gradient-based approaches. Let's talk about the algorithm that we're proposing. Essentially, we want to compute this thing here. This is the gradient of the t-SNE output with respect to one of its input features, in particular, a feature of a particular data point. And here we're going to just assume a one-dimensional t-SNE. You can see the manuscript for details for how to handle the 2D case. It doesn't really change the math very much. Basically, if you remember from before, the update formula, we have a gradient, the gradient of our embedded point with respect to the input is going to be the previous point, and then we're going to add another term which corresponds to the update. And we can use the chain rule to essentially derive this thing down here. And while it might look a little bit scary, this ends up not being too difficult to compute. Indeed, we can compute the derivative of q with respect to x and the derivative of phi with respect to x if we store the derivative of y with respect to x from the previous step. There's a few other points, but they're not too difficult to compute. And we derived similar equations using the Burns-Hut approximation of t-SNE, and we've created a Python package that can be installed using pip. And here's the QR code for a Python package. Now let's talk about some results. We performed some experiments on the following data sets. We have a synthetic data set, which is designed so that a small number of known features cause clustering patterns. We have an MNIST validation experiment, and we have a SARS-CoV-2 sequence data experiment, and this is a biologically relevant case study. Let's first talk about the MNIST dataset. When we run our t-SNE attribution method, we can look at areas in t-SNE space. Let's look at the area has all the 8 digits on it. We can see that if we zoom into each digit, we can look at the saliency map overlaid on each digit. And when we do that, we can see that the saliency map appears to be highlighting areas that are unusual on each digit. It's as if it were covering the idiosyncrasies per digit. We can also take a bird's eye view and take the average of the attributions averaged over each of the digit classes. And when we do that, we notice that each of the attributions sort of make a form of each digit, except that they highlight areas of the digit that appear to have a lot of variation. We can also see that in some cases, the attributions of some classes are very similar, and these correspond to digits that are very close by in t-SNE space. An example of this would be the 4 here and the 9 here. We clearly see that these two are quite similar, and indeed, 4 and 9 appear quite close together in t-SNE space. In general, it's very difficult to quantify the performance of feature attributions because we don't have ground truth. We designed a series of validation experiments to test the attributions without using ground truth comparisons. We did this because, despite qualitative results that seemed interesting, we wanted to be a bit more rigorous. We wanted to have a good quantitative assessment of what we were doing. We now describe the validation attribution experiment setup for MNIST. What we did was we took our digits, so we have an example here of a 4, and for each of these we took the attributions that we had computed using our t-SNE attribution method. And then we took the features that had the highest attributions and we applied a sort of corruption to them in order to corrupt the digit, and you can see that below. We only corrupted digits that had the highest attribution scores. And then we performed a t-SNE on the digits before and after the corruption. And of course, when you apply the corruption, the t-SNE becomes distorted, as you can see below, and the classes start to merge together and it becomes a big mess. What we did now is we took the Spearman correlation between the distances of the embeddings before and after corruption, the 10 nearest neighbor preservation. This is measuring if the 10 nearest neighbors of each point are the same before and after corruption, and the adjusted rand index, which we computed by performing a k-means clustering of the embeddings before and after the corruption and comparing how similar they are. And the idea here is that if the digits were very similar, if the corruption didn't do very much, then the digits would be very similar. And then the embeddings would be nearly similar and the scores would be low. And that would say that the attributions were not highlighting important features. But if the attributions were highlighting important features, then the embeddings after corruption would look very, very bad, and these scores would be very high, very low. We have here one of the experiments that we performed. In this case, we removed features based on the attribution values, but we moved the same features for every single digit in our data set. So in this sense, we had a global feature, we had a global attribution score. And we also, doing it this way, we could also compare it to other forms of unsupervised feature importance methods, including the Laplace score, and supervised methods such as the Fisher score. And you'll see on the top, we have the Spearman correlation, and on the bottom, we have the k nearest neighbors preservation score. And along the x-axis, we have the percentage of corruption that we did. So 2% all the way to 18%. And you can see that the score decreases as the percentage of corruption increases, which is expected. And in black, we have a random baseline, which is just basically removing features at random. And we did this a whole bunch of times. And we find that, of course, all the methods significantly outperform random removal. And surprisingly, the methods do very similarly on this task, despite the fact that each of these methods are coming from very different places. The gradient attribution is using the gradient of the t-SNE, and other methods like the Laplace score and Fisher score have their own theoretical framework. Now let's discuss the SARS-CoV-2 GAISE data. Sequences were collected from patients mostly by nasal pharyngeal swabs, as seen in the picture on the right. These were deposited to GLBIO, which is a global database of such SARS-CoV-2 sequences. A representative sample, selected by NextStrain, containing 2,374 sequences that were collected between December 2019 and January 2023. These sequences were accessed from GLBIO database on February 2023. This was the data that we used for our SARS-CoV-2 case study. Our pipeline looks something like this. We collected the samples from GLBIO using the NextStrain subsampling. We took the multiple sequence alignment file and we processed it, recoding it, removing problematic positions, and cleaning up the lineage metadata in order to get a World Health Organization lineage names. We then performed the PCA transformation on it and passed this into our TSNE as usual. But we also performed a interpretability analysis by using the gradients of the TSNE. And they result in something that looks kind of like on the right. This would represent the feature attribution scores that we collected and analyzed. We annotated the variants of concern using the naming conventions from the World Health Organization. For these lineages, we collected marker mutations from outbreak.info. And on the right, you can see a phylogenetic tree that was fitted to a subsample provided by NextStrain. So we plotted our TSNE embeddings, and what we can immediately see is that our TSNE generally clusters according to the variants of concern labels. For each lineage, we took the average of the attributions and plotted them against the mutation frequency within the lineage. We used dotted lines to display the threshold of significance, which we set to be the 90th percentile. We see that most markers have both high frequency and high attribution. Notice that our approach uniquely identified 3 markers versus, and this would not have been identified using an enrichment analysis. We're talking about these markers right here that have high attribution, but are below the 90th percentile threshold of frequency. Finally, we note that 25 markers were not identified using high attribution, but did have high frequency. We note that each of these were from lineages that were undersampled relative to the rest. In fact, these lineages had somewhere between 30 to 50 members of them, and this was significantly lower than the other ones. To conclude, we have developed an efficient method to generate explanations of TSNE embeddings. We developed a method that can validate their attributions. We hope that this method will inspire others to try their methodology and propose modifications. We note some caveats. TSNE and every other dimensionality reduction algorithm and every other machine learning tool can be misused. It is a mathematical fact that the embeddings will basically always produce distortion or artifacts. And you can see the Johnson, Lind, and Strauss lemma for that. Finally, we must be cautious about psychological factors like confirmation bias. I want to also mention that the same goes for interpretability methods. We have to be very careful when we use these methods. We have to be very mindful of these caveats. Next steps. We'd like to apply our method to other modalities. In particular, we're very interested in trying out single-cell transcriptomics data, where TSNE is very, very popular. We would like to apply gradient-based feature attribution to other dimensionality reduction algorithms. We'd like to adapt permutation-based approaches such as SHAP. There are many possible next steps. We're very excited to explore the strengths and limits of this method and to see where it could be used and how it can make your life easier. I want to say thank you to the funders of this research that are listed here. I also want to say a special thanks to all the people that helped me out along the way. My co-authors, Jean-Christophe and Raph, pictured here, and my advisors, Julie and Sebastian, pictured here. And to everybody else in the lab that helped me to get to this point. And thanks to you, the audience, for listening in. If you have any questions, please send them to mattcscicluna@gmail.com. Don't forget the C. And I will also be at GLBIO and giving a talk there, and happy to have any discussion about this. Thank you so much.